Simplified explanation. The intrinsic pathway of apoptosis is also called the mitochondrial pathway. It plays a role in normal development, like shaping organs in the embryo, and is also activated when cells are damaged or no longer needed. It is triggered in two situations. When growth signals are removed, for example, IL-2 is taken away after an immune response, causing immune cells to die. When cells are injured by things like radiation, toxins, or low oxygen. This pathway is controlled by a group of proteins called the BCL2 family. BAX and BOC help cause cell death. They are pro-apoptotic, bad for survival. BCL2 and BCLXL prevent cell death. They are anti-apoptotic, be clever, live. Here's how it works. BAX and BOC make holes in the mitochondrial membrane. This allows cytochrome C to escape into the cytoplasm. Cytochrome C then activates caspases, the enzymes that cause the cell to break apart. If BCL2 is overexpressed, it blocks cytochrome C from leaking out. This can prevent apoptosis and lead to uncontrolled cell survival, like in follicular lymphoma, which is linked to a genetic mutation called T1418. USMLE question 1. Which of the following proteins promotes apoptosis by forming pores in the mitochondrial membrane? A. BCL2. B. BCLXL. C. BOC. D. Cytochrome C. E. Caspase 3. Correct answer, C. BOC. Explanation. BOC and BAX are pro-apoptotic proteins. They create pores in the mitochondria, allowing cytochrome C to escape and trigger apoptosis. Why not the others? A. BCL2 blocks apoptosis by keeping the mitochondrial membrane sealed. B. BCLXL also prevents cell death. D. Cytochrome C helps activate caspases, but it does not form the pores. E. Caspase 3 is activated later in the process, after cytochrome C is released. USMLE question 2. A 45-year-old patient with follicular lymphoma has a mutation T1418, which leads to overexpression of BCL2. What is the consequence of this mutation? A. Increased mitochondrial pore formation. B. Increased activation of caspases. C. Inhibition of cytochrome C release. D. Enhanced apoptosis of tumor cells. E. Decrease in ATP production. Correct answer, C. Inhibition of cytochrome C release. Explanation. Overexpression of BCL2 keeps the mitochondrial membrane intact, preventing cytochrome C from escaping, which blocks apoptosis and allows tumor cells to survive. Why not the others? A. BCL2 prevents pore formation, not increases it. B. Caspases are not activated if cytochrome C is not released. D. This mutation prevents, not promotes, apoptosis. E. ATP production is not directly affected by BCL2 overexpression.